Today's lecture will be on the contraction of a muscle fiber, and particularly we'll be talking about this region called the neuromuscular junction. The neuromuscular junction is an intercellular connection where we see communication occurring between the nervous system and the skeletal muscle fiber. To understand this connection, we first need to understand some of the working organelles that are involved with the neuromuscular junction. As mentioned, the action potential or the signal will be coming down through the motor neuron into the first structure called the synaptic knob. This synaptic knob is located along the endomysium, and particularly it's located along the surface of the sarcolemma. Within the synaptic knob, we see cytoplasm that is containing mitochondria and vesicles filled with acetylcholine. These vesicles filled with acetylcholine are considered to be neurotransmitters, which are chemicals released by the neuron to change the permeability of the sarcolemma of the muscle fiber. The next region is this small region between the synaptic knob and the sarcolemma, which is called the synaptic cleft. This region separates the synaptic terminal of the neuron from the opposing sarcolemma surface. Within this, we see this region called a motor end plate. So this whole region here, including these areas that drop into the surface of the muscle fiber. This motor end plate contains membrane receptors or acetylcholine receptors. Also in the synaptic cleft, we find another neurotransmitter called acetylcholinesterase, which will help break down acetylcholine once the action potential makes its way into the sarcolemma of the muscle fiber. We'll be going through the steps of how a neuron stimulates the muscle fiber. As mentioned previous, this neuromuscular junction is going to be the connection between the nervous system and the muscle fiber. We will see an action potential that will be traveling down the motor neuron into the synaptic knob. And the first step after the arrival of the action potential, the electrical stimulus will stimulate these synaptic vesicles to release acetylcholine through exocytosis into the region of the synaptic cleft. Once the acetylcholine is released into the synaptic cleft, the acetylcholine will bind at the motor end plate at the acetylcholine receptor sites. Once acetylcholine binds to the motor end plate or the acetylcholine receptor sites, this will increase the membrane permeability throughout the sarcolemma. Once the permeability of the sarcolemma has reached, the action potential will travel across the full length of the sarcolemma, and sodium influx will generate this action potential. The action potential will then go down into the transverse tubule. While this is occurring, acetylcholinesterase will break the acetylcholine from the motor end plate. So as this action potential is traveling along the sarcolemma, going down through the transverse tubules, what we see is the beginning of the action potential, which is going to stimulate or trigger calcium to be released from the terminal cisternae. This calcium is then going to be released into the sarcoplasm. So once the calcium is released into the sarcoplasm, at the myofilament site, what we're going to see first is that the troponin, which is this blue, which is where the calcium is going to bind, these troponin are keeping the activation sites for the myosin heads inactive. So we would consider this slide to be at rest. In the next slide, what we're going to see is that the calcium here is going to bind to troponin. Once we see the binding of troponin, the troponin molecule will change the position rolling the tropomyosin strand. Again, the tropomyosin strand are these G-actin and F-actin strands, so shown here in red. So calcium will bind to troponin. Troponin will turn tropomyosin, which will then allow for the activation sites to be exposed. Once these activation sites are exposed, then we see the myosin head pivoting towards the G-actin or the tropomyosin, and we consider this to be the cross bridge. So the myosin or of the thick filaments are exposed to the activation site. At the same time, once the activation site or the cross bridge has been connected, we see that ATP will be utilized to cause the myosin heads to pull the thin filaments towards the M line. At this point, we would consider this muscle to be into a state of contraction. So this repeating action of attach, which means cross bridge, pivot, utilizing ATP, and then detaching and returning is considered to be one active contraction cycle. This cycle will continue, or contraction depends on three things: the duration of the stimulus of the action potential coming into the neuromuscular junction, the amount of calcium that's freely available to bind to troponin, and the availability of ATP to allow for the pivot or cross bridge and the contraction cycle to occur. So this is a picture of a motor unit. When we're looking at a motor unit is basically composed of a single motor neuron and all the muscle fibers it controls. So in this illustration, we see motor neuron number 1, which is shaded in blue, which is going to terminate, and it is going to control these muscle fibers here that are colored in gray. We see motor neuron number 1, which this action potential will travel down the axon, and then it will control all of the muscle fibers at the neuromuscular junction for all of the muscle fibers shaded in red. So this illustration is showing you an inverse relationship that exists between the size of the motor unit and the degree of control it's provided. So for instance, if we were looking at the eye, there'd be one motor neuron coming down that would be attached to, say, 4 different muscle fibers. If this was the quadricep muscle, we would see one motor neuron coming down and attaching to over 1,000 different types of muscle fibers. So when a motor neuron is stimulated, all of its fibers will contract at the same time. The total force exerted by each one of these muscle fibers is going to be dependent on the number of activated motor neurons or motor units.